Now, you have almost certainly heard of cancer. Now, why do you think here we say that cancer is a group of diseases that result in or from uncontrolled cell division? Well, let's think for a moment. Okay. We know that cancer cells divide in an uncontrolled fashion, and they crowd out normal cells. And because they crowd out normal cells, well, and they consume energy, of course, to divide, and they take up space as well, and so they hamper normal function. Well, wait a second. First of all, the diseases that result in uncontrolled cell division, yes, this is true because for a cell to become cancerous, it must have uncontrolled cell division. So the fact that somebody has cancer results in them having problems with uncontrolled cell division. But the actual symptoms of cancer, such as weight loss, organ failure, and so on, that's from the cells overgrowing and crowding out normal cells and eating up energy. So that's from uncontrolled cell division. So technically speaking, both are true. You have probably seen definitions of cancer that include either one, but in fact it's both. Just like many things are, in fact, why not both? Of course, this is rather less optimistic than most of those cases. Now, many cells, such as mature neurons in, say, the brain or the spinal cord, are not supposed to divide at all. Their specialized structures mean that for them to divide would require them lose their specialization, pretty much. And other cells, well, I mean, yes, okay, they're supposed to divide. For example, the gut lining is supposed to divide to replace the cells that are killed by exposure to stomach acid. Sure, fine, that's perfectly normal. Or cells that get scraped away by, say, fibers. By the way, reducing fiber in a diet is actually even worse for your gut because even though fibers can scrape away cells, your cells secrete mucus to make the fibers and everything else too travel along more smoothly. Which means that, yeah, okay, too much fiber is probably going to not be pleasant, but if you have too little fiber, you don't have enough mucus, and therefore the mass doesn't move along very quickly, it dries up, and can be very painful when being evicted. So, even for cells that are supposed to divide, they do so in a controlled way. For example, they get signals that, oh, the epithelial layers above us are getting thin. We should divide to bulk it up. Say, your skin. Your skin does not divide uncontrollably, even though people shed their entire external layer of skin about once a month or so. Of course, this depends on the part of the body. For example, your fingertips are basically guaranteed to weather away, i.e. be eroded, faster than, say, well, say some place that you don't touch, say your belly, or you does not rub against things very often. So, the controlled division of cells is controlled A, by nutrient supply. I mean, obviously, say if you have a cell, it is not supplied with enough food. It's not going to grow and divide. Of course not. It might even starve. Now, what else? There's internal signals such as, guys, the DNA is damaged. Let's try to fix it. And if we can't, well, the cell commits suicide to keep the rest of the organism healthy. But in cancer, well, the cell might not do this properly. And thus, it just keeps on dividing uncontrollably while accumulating DNA damage. Of course, sometimes cancer cells will self-destruct because they accumulated too much DNA damage and they just can't survive anymore. Sadly, this doesn't happen very often. Now, what else might there be? Well, okay, chemical signals. So, various hormones and other localized chemical signals. Hormones are, for the most part, when we advance further in biology, we use hormones as a word for things that spread throughout the body. Whereas there are localized signals. For example, if I get a paper cut, there are localized signals to the dermis, i.e. the layer under the epidermis, the living part of the skin, to divide further and fill up the gap. And of course, there's also other signals, such as released by mast cells to, say, histamines, to inflame the area so that more white blood cells can arrive and suppress any intrusion of bacteria or viruses. Now, the checkpoints in the cell cycle not only include nutrients and DNA damage, they also include, do I have the go-ahead to divide? For example, mature oocytes, i.e. the precursors to egg cells, they actually suspend before meiosis quite begins. And they remain suspended that way from fetal development up until that ovum, well, that oocyte decides to develop. So until that particular menstrual cycle. So for many dozens of years if need be. Now, this arrest of the cell cycle, which is pretty standard, even though many of these cells, which are often in the quiescent, i.e. doing nothing, so to speak, except functioning normally, they are doing nothing in terms of the cell cycle. That doesn't mean they aren't functioning. Your neurons are all in G0. If they didn't function, well, you get the idea. Now, this is not the case among cancer cells. They have basically no checkpoints left that actually work, and the suicide signals for DNA damage do not work, and they are not well regulated by chemical signals, be they hormones or local signals. Now, cancer cells, well, they aren't the only type of cells that actually do this. There are non-cancerous cells which are still tumor forming. A tumor is basically a mass of cells, a mostly solid mass, that keeps on growing and dividing without any obvious function. So they don't actually do anything useful. So they just take up room and nutrients, but they don't do anything useful. Well, now what's the difference between a benign tumor and a malignant tumor? Well, a benign tumor stays in one mass. It doesn't spread around. Now, typically, this means that it has less mutations and less DNA damage than a malignant tumor, because it takes a few mutations for the cells to detach from the substrate and then go wandering about throughout the body, establishing new tumors. So a benign tumor needs a few more mutations to become malignant. A malignant tumor will interfere with the surrounding cells other than by physically crowding them. So a malignant tumor will spread itself into nearby tissues. In fact, malignancy was the reason why people like Hippocrates characterized cancer as carcinos. Carcinos is actually the Greek term for crab, which means, well, wait a second. Let's think about how this works. Okay, a crab has a hard central body, and it has tendrils that spread out. Okay, so legs and claws technically, but you can visualize them as tendrils. Wait a second. A malignant tumor has a solid body, like other tumors. So it's a mass of non-functioning cells. It's pretty solid. So a solid, hard body. And then it has branches that stick out into surrounding tissues. Hence the name carcinos and cancer. Cancer is the actual Roman term because they took all the Greek books and so on and they translated them into Latin. So cancer is a Latin word for crab. You might have heard of the constellation of Cancer or Tropic of Cancer. That's because the Tropic of Cancer crosses through the constellation Cancer. Now, this ended up resulting in most Latin-influenced cultures adopting their variant term for crab for cancer. If I recall correctly, at least Poland and I think Russia actually use their local term for crab for cancer, or a term directly derived thereof. Now, metastasis. This is the characteristic that that for the most part defines the difference between a malignant and a benign tumor. For the most part, a tumor can be considered benign until it starts to spread. And by spread, we mean some of the cells come off, enter the bloodstream. By the way, this migration takes quite a number of mutations because the default for human cells is not anything like this. Except leukemia, but that's another matter. So this could be, say, in the lungs, or in the liver, or gut, or whatever. This is unlikely to be in the heart. Why? Because cardiac muscles do not divide after you reach adulthood, at least. So once the heart stops growing, cardiac muscle cells will basically stop dividing. So with very few cell divisions, what does this mean? This means not much time for mutations to build up. Or not very many chances, because mutations build up every time you replicate. It doesn't matter how long you took to replicate, say 3 months between cell divisions, or 3 days, it doesn't matter. What matters is the number of replications the cells have undergone. By the way, this is part of why most mutations in the human lineage come from males, because sperm cells are constantly being produced, and the way they are being produced means that while the average egg cell begins with a cell that has undergone, say, 50 or so, some numbers in the tens of cell divisions, the average sperm cell comes from a cell that has undergone hundreds of cell divisions. So, more chances for mutations to happen and pile up. So this could be, well, from any of these tissues. So it enters the blood, and then it attaches to the blood cell wall somewhere else, and it forms a metastasis. It may be of the same organ type or different organ types. Now, so those are the identifying traits of cancer. That it spreads, unlike a benign tumor which stays in one place, even though it is useless and it might keep growing. This is part of why many people bother to have benign tumors removed, because just in case some cells in there actually turn cancerous, they don't want a diagnosis to come too late. The earlier a cancer is caught, the better the prognosis. So, causes of cancer. Well, DNA replication is pretty good, but mistakes do happen. It's not perfect. Therefore, as mutations accumulate, more and more of the cell cycle control genes can get knocked out or otherwise be activated at the wrong times, and, well, the cell could end up dividing uncontrollably, and thus tumor. And then a few more changes to mobilize the cell, make the cell able to move into the bloodstream and spread, and there you have cancer. Now, we note that technically carcinogens listed as environmental factors that cause cancer overlaps greatly with mutagen. Mutagens cause mutations. I mean, we know that genesis means to make something, so mutagens generate mutations. I mean, this makes sense, right? Just as carcinogens generate carcinos, i.e. cancer. Well, carcinogens can be chemicals. For example, tobacco smoke contains a very complex mixture of many different carcinogenic chemicals. And of course, stuff like carbon monoxide. So anybody who smokes basically has long-term chronic carbon monoxide poisoning, which, well, is not good for blood's oxygen supply to, say, muscles. Now, carcinogens can be physical, such as ultraviolet light. Now, ultraviolet light is capable of inducing thymines, the base thymine in DNA. There are 4 bases. So, A, adenine is paired with thymine, and guanine is paired with cytosine. So, thymine under ultraviolet light will form dimers. So two adjacent thymines will end up cross-bonding with one another, which produces a kink in the DNA. And if this is not repaired before the DNA replication machinery passes over it, well, it could mess it up. Now, some viruses also put parts of themselves in the host genome or modify the host genome in some way. If they put parts of themselves into the host genome, well, there are several types. There are DNA viruses, which could just insert themselves directly, or it could be an RNA virus that undergoes retrotranscription. So it goes from RNA to DNA and then inserts that part of itself into the host genome, such as HIV or, in this case, HPV. HPV, human papillomavirus, is responsible for many, many cervical cancer deaths each year. Now, hepatitis B is a common illness that can be obtained from eating at restaurants if you are not vaccinated. Of course, these restaurants have to be of questionable sanitary condition, but the point is that it is often obtained from eating at restaurants, which is why I don't like visiting restaurants, but that's not the point. So the point here is that hepatitis B can cause liver cancer, which is one of the more difficult cancers to treat and detect initially. Why initially? Well, okay, skin cancer, usually it's pretty obvious if it's in a relatively visible location. Now, even benign skin tumors in relatively visible locations, such as this mole here, well, even benign tumors can cause a great deal of alarm and cause people to get them removed as soon as possible, just in case something happens. For the most part, though, people leave moles alone because potential infection and such in hospital environments tend to be more dangerous than, well, the moles. Now, liver cancer or any other internal organ cancers tend to be a little bit harder to detect because they're not blatantly visible, or alternatively, you can't just feel them. Hence the self-checking for breast cancer. Now, cancer can also be hereditary. Why? Wait a second. If I have normally, okay, 2 sets of chromosomes, 2 pairs, okay, well, 2 sets of chromosomes, that means I have 2 sets of gene repair machinery. Wait a second. I also have 2 sets of cell cycle control machinery. So wait a second. What happens if one set is mutated somewhere? So it's already broken. Okay, the other set is still working. All right, it could be enough. It might not be enough. But for the most part, if it's a live birth, for the most part, that set is enough. And, well, what happens if this set breaks? Clearly, you have a much higher risk for cancer if you have a family history of cancer because of, well, inherited cell cycle or DNA repair machinery issues. Smoking is probably one of the major causes of cancer deaths, other than obesity. Now, the carcinogens in tobacco smoke affect all of these organs. Actually, they probably affect your skin as well, because it is known that people who smoke will have skin that smells faintly of tobacco. Now, nicotine is powerfully addictive. It is probably the most addictive drug. In fact, it is often considered more addictive than heroin. But, well, caffeine is actually second place, considered by many. You will notice that many young people will try out, say, a certain type of weed that I'm sure we are all familiar with, or at least all know of. I mean, I have never even figured out how to identify the smell of it, but I do know that many young people have tried marijuana, and, well, most of them don't try it again. How many young people do you know who have tried drinking coffee and managed to quit? But of course, coffee is a richer industry which is worth more money, and therefore can invest more money in advertising and such. So nobody really cares about coffee being harmful to people or addictive. Because, well, it's not as harmful as, say, tobacco. Tobacco smoke, which is legal because they have enough money to throw at things to keep it legal. Tobacco smoke, first of all, nicotine is known to cause deaths by overdose. There has been no medical effort that has managed to successfully kill an animal, a large mammal, such as a dog, with overdose of marijuana. Other than from smoke inhalation or similar, it is pretty much impossible to overdose on at least tetrahydrocannabinol, which is kind of interesting. But of course, the banning of marijuana back in the early 1900s, after so many years of use as a painkiller in herbal medicines, well, do you know how that started? That actually started with drug companies developing chemical painkillers. So what they wanted was to ban the natural painkiller so that they could sell theirs for more money. And thus the lobbying efforts began. Well, you should be interested to know that while the vast majority of lung cancer deaths are by smokers, that for quite a while people did not think that tobacco smoke was harmful. In fact, even in the 1950s, the tobacco industry paid a bunch of doctors and scientists to convince people that tobacco smoke was not harmful. And eventually, well, their fraud was, or whatever you call it, was exposed. Their lies. Now, we do note that lung cancer is one of the more common types of cancer, and that it's relatively difficult to treat. And of course, 9 out of 10 or so cases are due to smoking. Now, wait a second. If cancer is so dangerous, then of course we've developed many methods to try to detect it before it becomes untreatable. Now, screening involves checking even if there are no symptoms.